Hey class, this is chapter 13, glucose metabolism. We're going to go over this one fairly quickly. Now, starting with this, glucose occupies a central position in the metabolism of most cells. This is a major source of metabolic energy. In some cells, it's the only source, and it provides the precursors for synthesis of other biomolecules. Now, glucose is stored in polymeric form as starch in plants and glycogen in animals. Now, we discussed this in chapter 11. The breakdown of these polymers provide glucose monomers that can be catabolized to release energy. Now, what we're going to look at is this chapter that is the major metabolic pathways that include glucose, the conversion of it from monosaccharide glucose with glycogen, degradation of glucose, and all of that. So that is what we're going to look at in this chapter. Glycolysis is the conversion of glucose to pyruvate. This is a good place to begin a study of metabolic pathways. This has concluded with a lot of years of research. We know how to deal with this. We all have also learned that glycolysis, along with other metabolic pathways, have certain properties. So this is what we're gonna talk about in this section. We're gonna talk about each step of the pathway is catalyzed by a distinct enzyme. The free energy is consumed or released in certain reactions, is transferred by molecules such as ATP and NADH, and then the rate of the pathway can be controlled by altering the activity of individual enzymes. What you see here in the figure is glucose metabolism in context. You have biopolymers, polysaccharides. Glycogen is degraded to glucose, which is then catabolized by glycolytic pathways. The three carbon intermediate pervolate. Um, so that's what you see in number two. Then number three, gluconeogenesis is the pathway for the synthesis of glucose from smaller precursors. Glucose can then be reincorporated into glycogen, and then the conversion of glucose to ribose is a component of the nucleotides, and that is not shown in the diagram. Now we're going to go through the phases of glycolysis. And so now reactions 1 through 5 are considered the energy investment phase of glycolysis. So the first 5 reaction is a preparatory phase for the second, and then the second phase is energy producing. So this is the energy investment phase. So step 1, what happens is hexokinase is transferred to a phosphoryl group from ATP to the C6OH group of glucose to form glucose 6 phosphate. So that's what happens in this part right here. So this is the first 5 steps. Then you got phosphoglucose isomerase, and that's the second reaction of glycolysis when the glucose 6 phosphate is converted to fructose 6 phosphate. Then what happens after that, you got phosphofructokinase, and then what happens is fructose 6 phosphate yields fructose 1,6 biphosphate. Then in step 4, you got aldolase, and then it converts the hexose fructose 1,6 biophosphate to 2 3-carbon molecules, of which each one of them bears a phosphate group. Then you got the triose phosphate isomerase. Now these products of the aldolase reaction are both phosphorylated 3 carbon compounds, but only one of them, and that's the glyceride 3-phosphate, proceeds through the remainder of the path. Dihydroxyacetone phosphate is converted to um, glyceraldehyde 3-phosphate by triose phosphate isomerase. So this is the first 5 steps of glycolysis. Now we are dealing with the last 5 steps of glycolysis, and this is considered the energy phase. Now it's what they consider the energy payoff of glycolysis. So step 6 deals with glyceride aldehyde 3-phosphate dehydrogenase. Um, that's what happens is both oxidized and phosphorylated. So it's oxidized, phosphorylated, and then you get phosphoglycerate kinase. And then when you get that, that's an alkyl phosphate. And so that is then converted. And then once that converted, in the next reaction, you got phosphoglycerate mutate is converted to what happened is 2-phosphoglycerate. So that happens in that, and then step 9, you have your enolase, and enolase catalyzes a dehydration reaction, and in this case, water is eliminated. And then step 10, you got pyruvate kinase. And this is the 10th reaction of the glycolysis right there. Now, when you are dealing with that, you're going to be dealing with tautonomers. And tautonomers really deals with the isomerization through the shift of a hydrogen atom. We'll get started on that on the next slide. So to break all of this down, they got the hexokinase reaction. I just talked to this, the phycoglucanase isomerase reaction right here. The phosphofructanase, that's where if we go back, and I can't go back right now, but you have basically all of these. So this is each step. Now each step of all of these are very important, because I'm going to want you guys on the test to be able to explain each step, because this last test y'all going to have a part 1 and a part 2 where you have a little bit of math, and then you go through this and go through each one of the steps. Now, in this slide, the fructose 2,6 biphosphate is the most potent activator of the phosphofructanase in mammals. So this is important when this is converted. Other molecules activate or inhibit phosphofructanase. So glucose, if you're looking at this and you're trying to get down to pyruvate, you can have other things that can activate it or inhibit it. And so these are just some examples here that you actually see. So you have the aldolase reaction, and you take that 1,6 biphosphate and then you convert it. And then this is the aldolase mechanism in detail. The fructose 1,6 biphosphate shown in this linear form, it binds to the enzyme, and that is what you actually see. And then 2, the active site LYS residue reacts with the substrate carbonyl group to form that SCIF base. Now, we've been talking about this a little bit in organic too. So, so you see this, that's what we're talking about with the SCIF bases in organic too. Now then, the ASP side chain acting as a base accepts a proton from the substrate, and the bond between the C3 and the C4 breaks. then releasing the first product. It releases that aldehyde. The reaction is facilitated by a protonated skiff base, which is better, which is a better electron withdrawing group than the carbonyl group that was originally at C2. The AsP residue give off a proton for the remainder of the substrate, reforming that skiff base. It reforms another skiff base. So this keeps going on and on and on. And then the skiff base is hydrolyzed, releasing the second part and regenerating the enzyme. So what happens is it's going to keep regenerating. It's going to keep regenerating because of that protonation. And like I said, we've been talking about this in organic chem. So you actually see this right here. Okay, what you see here is step 5, the triophosphate isomerase. And what here you get the dihydroxyacetone phosphate, and it is then converted, and you use the triophosphate isomerase to convert it. Now, this is the last step in this part of it before we go into the energy payoff. So this last step is just the preparation phase. Now, triose phosphate isomerase is the catalytically perfect enzyme. So that's why it is often done last, because it is perfect. What do they mean by that? It has one loop of the protein having a residues of 166 to 176. That is highlighted in the green. When the substrate binds to the enzyme, the loop closes over the active site to stabilize the reaction's transition state. In this model, the transition state analog to phosphoglycolate, and that's orange, that's in the little orange right there on part B, occupies the active site. Triose phosphate isomerase is actually a homodimer. It's the only subunit pictured here. It's only one of them that you see here. Now, when we get to step 6 through 10, this is the energy payoff. So what we're seeing is that aldehyde phosphate is getting converted. So we're going to use the glycidylaldehyde 3-phosphate. It's getting converted to 1,3-biphosphoglycerate. So that is what's happening there. So that reaction is getting converted, and this is called the GAP dehydrogenase reaction. And the GAP dehydrogenase mechanism, so the GAP binds to the enzyme who active site already have NAD+. Then the SH group of the active site, CYS residues form that covalent bond to GAP. And then NAD plus oxidizes the substrate, forming that thioester intermediate. And then that NADH exits and replaced by another molecule of NAD plus. P sub I enters that active site. Then the inorganic phosphate attacks that thioester, forming 1,3-biophosphate glycerate and regenerating the enzyme. Now, once this is done, that 1,3-biphosphoglycerate is then converted, and so this is where you have the phosphoglycerate kinase reaction. And then step 8, this is where the mutase take over. So you go from 3-phosphoglycerate to 2-phosphoglycerate. So that's what it's doing. It's moving. And this isomerization of the 3-phosphoglycerate occurs via an active site His residue. So this is what you see going on right here. And then step 9 is your enolase reaction. And so the enolase catalyzes catalyzes a dehydration reaction, and this is basically where water is eliminated. And then step 10, this is where you get your pyruvate. And so this is the pyruvate kinase reaction. So this is the 10th reaction, and it converts to pyruvate and transfer the phosphorus group to ADP to produce ATP. That is what happens. Now, what happens is with all of this, you got all of these free energy changes. So the three steps have large negative values of delta G, meaning that that's a spontaneous reaction. The remaining steps happen near equilibrium. That's where delta G is almost or really close to zero. The height of each step corresponds to its delta G values in the heart muscle, and the nerves correspond to the glycolytic enzyme. Keep in mind, delta G is going to vary between the tissues, and so it's going to vary slightly. Now, what happens to the pyruvate? What happens to it? That is a good question. It can convert to other substances. Okay, in here, it can convert to a 2-carbon acetyl group linked to the carrier coenzyme A. And then you can have acetyl-CoA may be further broken down by the citric acid cycle or used to synthesize fatty acids. In muscle, however, pyruvate is reduced to lactate to regenerate NAD plus for glycolysis. Yeast degrade pyruvate to ethanol and CO2. Pyruvate can also be carboxylated and to produce 4-carbon oxaloacetate. So a lot of things can happen to pyruvate. Now, sometimes during exercise, pyruvate can temporarily convert to lactate as well. So it can actually do that. So you see here you got pyruvate and then NADH, and then it does lactase dehydrogenase, and then you get lactate. Now, what else can happen is you can have fermentation. Now, organisms such as yeast growing on a different condition can regenerate NAD plus by producing alcohol. Now, Louis Pasteur called this process fermentation, and that is what happened. And basically meaning life without air, although these also ferment sugars in the presence of O2. Now, um, let's talk about it. You can look at this and be like, for one, alcoholic fermentation is a 2-step process. And what happens is the pyruvate decarboxylate, and that's an enzyme not present in animals, cannibalizes the removal of the carboxylic group to produce acetaldehyde. Next, alcohol dehydrogenase reduces aldehyde to alcohol. Now, what else can happen is that pyruvate can be further oxidized. Um, in the other one, ethanol is considered a waste product of sugar metabolism, is accumulation of toxic to other organisms, including that it also produces yeast. This is the one reason why alcohol content of yeast fermented beverages such as wine is only limited to about 13%. Hard liquor must be distilled to increase the ethanol content. Now, what you see here is glycolysis is an oxidative pathway. Its end product, pyruvate, is still relatively a reduced molecule. The first catabolism of pyruvate begins with decarboxylation to form a 2-carbon acetyl group linked to coenzyme A. And this is what you see here. Okay, this is just a chart that is showing the standard free energy changes for glucose catabolism. And so you see this here. And free energy is very important because we have to understand whether or not, when you're looking at free energy, do these reactions spontaneously happen, or do we need to add a catalyst to it? Do the enzymes, how do they make the reaction go faster? So how everything breaks down in our body. Thinking about even when, breaking down when we take multivitamins, when we take certain medicines, all of this, the enzymes that we use have to break all of this down. And so this is very important, especially for people who are going into medicine, because what we have to understand is, let's just say, for instance, we're talking about the coronavirus. This is the big thing. This is why we're at home. And one group of individuals that this really affects is people who have diabetes. Understanding how glucose breaks down is understanding how people are affected that have certain medical conditions like diabetes. Now, pyruvate is a precursor of oxaloacetate, so we was talking about that earlier. So this is what you see here. Now, pyruvate carboxylate use biotin as a cofactor. So you can see that, and that can happen as well. Now, how does this mechanism actually work? Now, CO2 is in a bicarbonate. It reacts with the ATP, that some of the free energy is released in the removal of that ATP phosphorus group. And when it's conserved, it activates it. And then, like ATP, that carboxyphosphate releases a larger amount of free energy when its phosphorus group is liberated. This free energy drives the carboxylation of biotin. And when that happens, the enzyme abstracts protons from abstracts a proton per pyruvate, and it forms that carbocation. And so when it do that, it attacks that carbonyl group, and it generates that acetate, that oxyacetate. So that is what happens. And so with that, we're going to get into now gluconeogenesis. And we want to look at the substrates, the products, and the reaction of gluconeogenesis. And so this pathway, which is also occurs to a limited extent in the kidneys, operates when the liver supply of glycogen is exhausted. Certain tissues, such as the central nervous system, red blood cells, will burn glucose as their primary metabolic function relies on the liver to supply them with newly synthesized glucose. Now, gluconeogenesis is considered sometimes to be the reversal of glycolysis, that is, the conversion of 2 molecules of pyruvate to 1 molecule of glucose. So we're going to take a look at that and talk about that in this section. So this picture right here just gives you the reactions of gluconeogenesis. The pathways use 7 glycolytic enzymes that catalyze reversible reaction. 3 irreversible reactions of glycolysis are bypassed in glucogenesis by 4 enzymes that are highlighted in blue. So that is what happens. So the glucose 6-phosphate, the fructobiphosphate, um, the pyruvate carboxygenase, and then the pyruvate carboxylase is there. Okay, on this slide you see pyruvate is converted to phosphoronopyruvate in 2 steps. So this is what you see here on this slide. Now gluconeogenesis is regulated by fructose biphosphate step, and this is what you see here. What you see here is the fructose 6-phosphate is a potent inhibitor of fructose biphosphate, which catalyzes the opposing gluconeogenic reaction. Now, what happens to glycosynthesis and degradation? So, with that, what we have to look at in animals, dietary glucose and the glucose produced by gluconeogenesis are stored in the liver and other tissues as glycogen. Later, glucose units can be removed from glycogen polymer by phosphorylases. Because glycogen degradation is thermodynamically spontaneous, glycogen synthesis requires the input of free energy. The two opposing pathways use different set of enzymes so that each process can be thermodynamically favorable under cellular conditions. Now, glycogen is a polymer of glucose-1-phosphate monomers. That is one thing that you have to understand. So this is a monosaccharide unit that is incorporated into glycogen, and that's the glucose-1-phosphate. And it is produced from glucose-6-phosphate by the action of this enzyme here. Now, with this, glycogen synthesis consumes the free energy of UTP to form UDP glucose. Like GTP, UTP is energetically equivalent to ATP. And this is what you see on this slide. Now, glycogen synthase adds glucose to extend the glycogen polymer. So that is what happens. So in this part right here, that's what you see. So what you see here is, let's see here. Cleavage of a phosphor and hydride bond make the formation of UDP glucose as exogonic irreversible process. That is, PP sub I hydrolysis drives a reaction that would otherwise be near equilibrium. The hydrolysis of PP sub I by inorganic phosphoratase is a common strategy in biosynthetic reaction. You'll see this reaction again in other polymers, DNA, RNA, polypeptides. So glycogenolysis, that is what you see. Now, the last thing that we're going to look at is the pentose phosphate pathway. Now, that converts the glucose 6-phosphate to ribose 5-phosphate. It's an oxidative pathway that occurs in all cells. But unlike glycolysis, the pentose phosphate pathway generates NADPH rather than NADH. The two cofactors are not interchangeably and are easily distinguished by degenerate enzymes, which generally use NAD plus and biosynthetic enzymes, which generally use NADP+. The pentose phosphate pathway is by no means a minor feature of the glucose metabolism. As much as 30% of glucose in the liver may be catabolized by pentose phosphate pathway. This pathway can be divided into two phases: a series of oxidative reactions followed by a series of reversible interconversion reactions. Now, what you see here, the oxidative reaction of the pentose phosphate pathway produced NADPH. Now, this is the starting point of the pathway is glucose 6-phosphate. So that's the starting point. It's derived from free glucose from the glucose 1-phosphate produced by glycogen phosphorylase or from gluconeogenesis. So that's what you see here. And then here you have your 6-phosphogluconate production. And so with this right here, the lactone intermediate is hydrolyzed to 6-phosphogluconate by the action of 6-phosphogluconatase. Although this reaction can also occur in the absence of an enzyme. So this is what you see in here. The third step of the pentose phosphate pathway involves oxidative decarboxylation. So you see the 6-phosphogluconate, and then it reacts, and then what you get is the ribulose 5-phosphate. And then the ribose 5-phosphate is a precursor of the ribose unit of the nucleotides. And the ribonucleotide reduces and converts ribose to deoxyribose. So that's what you're seeing here. And so you have rearrangement of those products in the pentose phosphate pathway. And then right here, what you see is a summary of the glucose mechanisms. So what you see here is the diagram includes the pathways of glycogen synthesis, degradation, glycolysis, gluconeogenesis, and the pentophosphate pathway. The dotted line are used where the individual reactions are not shown. Field gold symbols indicate ADP production. Shadow gold symbols indicate ATP consumption. Field and shadow red symbols represent the production and consumption of reduced cofactors NADH and NADPH. Now, what about disorders of carbohydrate metabolism? What kind of disorders that you see? You can see enzyme deficiency. So that's what you see here in the chart. You got glycogen storage diseases that can happen. So there are different types that can happen.